So I'm sharing my screen in order to basically go through two sessions first, because it is the first session for a lot of people who have not basically who were not present during Friday's lecture, or they have enrolled later, right? So I will just quickly go through what we have done in the previous session, right? I will not uh, explain all the concepts right now. There is a separate recording for that that is available. Why I'll quickly go through what we have already done is so that whoever has missed that session can get a context of what we are dealing with, what we are studying, because we are picking up from where we have left in the previous session, right? So that it will be more contextually relevant for you to attend the session, I will quickly give a recap, right? And I think we can make it a habit of doing a quick recap of the previous session. Although not the entire slide, maybe I will sum it up as points from next class onwards. But over here, since it is mainly historical based, so I thought maybe looking at the slides will help you recall better, right? Because it is more memory based than actual logic based, right? So you would remember what has been done, but who has done that particular stuff, you might not recall, right? Just by the pointers. So, for that part, we have kept this session, this quick recap. So, I have kept these slides also. As for a formal introduction of myself, which I think I should have done first, I am Arvind Kaur. I am currently a PhD scholar at Department of Biological Sciences and Bioengineering at IIT Kanpur. And like this is my area of research also. So, I'm working on the neurobiology of diseases. Um, that is my research area. I am working on neurodegeneration and how there is uh, a presence of, or could be a presence of blood-brain barrier defects in these neurodegeneration. So that research specific part, I might take it up in another lecture of mine, for in another module of mine. For this particular module, that is basics of neurobiology, we will deal with um, uh, the basic concepts pertaining to neuroscience and neurobiology, right? So in the first lecture, we quickly went through what is neuroscience, what is neurobiology, right? So both pertain to study of the nervous system. The major difference between both of them is that neuroscience is a multidisciplinary field, right? Not only does it encompass physiology, anatomy, molecular biology, developmental biology, cytology, which are multiple fields of biology themselves. It also incorporates subjects like psychology, physics, computer science, chemistry, medicine, statistics, and mathematical modeling, right? To understand what the brain is, how it is functioning, how is cognition developing, etc. Right? That is actually the major difference between neuroscience and neurobiology. Neurobiology deals mainly with the part of the neuroscience. It can be a sub part of neuroscience, right? Because it deals with the part of neuroscience that has to do with anatomy, physiology, morphology, cell and molecular biology, etc. Right? So it is the study of brain and nervous system, and it is basically where we are studying the cells and the tissues that are generating certain movements, making certain decisions, helping us learning movement, consolidating memories, etc. Right? Then we came to the very famous discussion that has always been a part of neuroscience, which is the mind and the brain, right? So we did discuss about how mind is an intangible part, and it is like a concept which doesn't exist as a physical entity, but it is there, right? It is the process through which our consciousness and thought are existing, right? While brain is a tangible part, it is like the machinery which is responsible for this. Thought and consciousness, the working of it is happening in the brain itself, right? So we compared brain to the and the neurons in it to the Milky Way, right? And we know that brain is tangible, that is, we can touch and feel it, while the mind is intangible, right? So if we consider the brain as the hardware, right, then we can consider the mind to be the software, right? In case of this computer systems also, software is an entity that you cannot physically feel, right? But it is there, it is what is computing everything for you, right? Similar is the case with brain and mind, right? So if brain is the hardware, mind is the software, right, where all the processing is occurring. And when we talk about the mind, we talk about realm of consciousness, thoughts, emotions, perception, etc. Right? It is a part of us that experiences the world, right? And it helps us solve complex problems. It is what part of us helps, helps us dream, etc. Okay. However, mind is intangible. We have already gone through this. So, what is the difference that exists between mind and brain? Right. The first is the mind is tangible, while a brain is tangible, while the mind is abstract. Right. It doesn't exist in as an entity. Right. So brain, the activity of brain can be observed and measured, right? So it is very subjective. So it is very objective because it can be objectively measured, but the contents of mind are are very subjective, right? So, for instance, I gave you an example of a pouch which had red color in it, right? So I told you that the perception of red that I will have very will be very different from the perception of red that you will have, right? So up to that level, the mind is subjective, right? And only my mind knows what exact color of red I am seeing, and only your mind knows what exact color of red you are seeing, right? We cannot explain it in terms of quantifiable techniques, right? So that is what is subjectivity. Then again, measurability versus observability, right? So the uh, workings of the brain are measurable, while workings of the mind can only be observable, right? You can observe that, okay, uh, a person solving problems, but you cannot exactly say how or what part of the brain is actually solving that problem, right? Then the brain's primary job is to keep you alive and functioning, right? So it is basically that system, like the CPU of the computer, which is responsible for the computer to remain on, right? The mind, however, deals with higher functions like reasoning, creativity, and self-awareness, right? That is not a function of the brain. The brain's parts help achieve those functions, but it is a function of the mind, right? Also, brain is ages over time. The cells will die, they will age, there will be cognitive decline that is happening because of the neurons dying. The mind, however, will continue to develop and grow, right? Due to experiences that we gain, wisdom that we gain, new skills that we learn, right? So we pondered more upon this relationship that although they are distinct, they are very intimately related, right? And this relationship is all is what helps our body and our self function, right? So your thoughts and emotions can actually change your brain structure, right? So if we are thinking something, or if we are perceiving something and it is becoming a memory, that is because the structure of our brains, the connection between our neurons is getting altered, right? So there is a connection between brain and mind, right? It does exist in some ways, right? So the current focus of our study throughout this lecture will be the unified theory. What is the unified theory? It is the unification of the study of behavior, that is the science of the mind, and neural science, which is the science of the brain, right? So we will take a unified approach in which mind and body are not viewed as separate entities, but uh, they rest on the view that behavior is a result of the brain function, right? So that is what our approach will be while we are taking on this subject, right? Also, brain processes underlie not only simple motor behavior such as walking and eating, but also complex cognitive acts and behavior such as thinking, speaking, creating works of art. Some of these examples we are going to deal with in today's lectures. It will basically consist of this create speaking and thinking part, right? Case studies related to that, so that we are going to take up today, right? So you will get a deeper understanding about this, right? So then we came to few questions that are still prevalent and open questions that neuroscientists worldwide have been trying to answer, right? And we then came upon the historical perspective, right? So, how the understanding of brain evolved over time, right? First, we started with Galen. So, who is one of the oldest people whose record is there? He was a Greek physician in the second century itself. And what he believed was that brain was the seat of mind. There are ventricles that are present in the brain, which are basically the hollow spaces of the brain. We will, once we look at the brain's anatomy, I will show you where the ventricles are, right? However, what he believed was that the nerves that are arising from the brain, they contain some fluid that is secreted by the brain, which they are taking to the other body parts, which is how brain is controlling the other parts of the body, right? And it was uh, like in second century AD, it was a good, rationale at that time because blood vessels also do the same kind of things, right? They look also very similar to the nerves, right? So, it, uh, like when this was thought of, it was a good rationale, right? However, we know now that that is not true, right? Nerves are not conducting vessels of any kind, right? They work in a much different fashion, right? Then we studied about the Italian scientist Camillo Golgi and Spanish Spaniard Santiago Ramon y Cajal, right? And we learned how Golgi developed the silver staining technique, right, which is also known as the Golgi-Cox staining, through which selectively the neurons are identifiable under a microscope when you take a brain slice and you stain it with the silver nitrate or silver salts, one of which is silver nitrate salts, right? And it looks somewhat like this, right? However, we also studied that uh, Golgi believed that in the brain, all the neurons were a part of one single unified network, right? So every neuron was connected and it was forming a one single unified network. And this was the reticular theory, right? So he supported this theory. However, Ramon Cajal, who was a Spanish fellow, he used the same technique that was developed by Golgi. He, stained the brain slices and he made very intricate drawings of the brain regions, right? After he made these intricate drawings, he basically would observe that there are individual nerve cells or neurons that are present, right? So he basically formulated the neuron doctrine, right? And this neuron doctrine said that individual neurons are the elementary building blocks of and signaling elements of the nervous system. And this stands true even today, right? And this was a representation of a few of the hand drawings that were done by Cajal. So at that time, actually, microscopes were available, but cameras attached to microscopes had not been developed as yet, right? So what Cajal used to do was he had stained various sections of a brain and he used to keep these sections under a microscope, take a look at them, keep a copy a drawing pad, and he used to draw whatever he used to see on the slides. See, all these are hand drawings done by Kahat, right? Uh, log of it is also present in this beautiful book, which is known as The Beautiful Brain, which has a compilation of the drawings of Santiago Ramón y Cajal, right? Yeah. So next, this neuron doctrine was supported by multiple scientists, one of which was Ross Harrison, right? He basically showed that dendrites and axons grow from the cell body, right? Even if these are separated from other neurons or separated from other tissues, they will still grow from one cell body, right? So that proves that it doesn't require that network to grow, right? It is not growing from one neuron, another is getting formed, and then another is getting formed. That is not what is happening. And he also basically confirmed the suggestion that tip of axon gives rise to an expansion, which is growth cone, which leads to developing axon to its target, right? So we have discussed about this in detail last time. So I'm just speeding through the slides, right? Then Sanford Pillay again was another scientist who provided definite proof of neuron doctrine, right? What he did was he demonstrated existence of synapses, right? So one, what is synapse? Synapse is a gap between two neurons, right? Gap through which the chemical messengers or electrical impulses are transmitted from one neuron to another, right? So that conduction of signal can take place or nerve impulse can take place, right? So these synapses, because they are gaps, right? They can definitely prove that this is not a continuous network, right? There are separate neurons that are existing that are forming a kind of network, right? It is not arising from one neuron. Then Luigi Galvani, who was an Italian physician and a physicist, discovered that muscle and nerve cells produce electricity, right? So what Galvani did, there was a very famous experiment that he did with the legs of a frog who had been dissected, right? So this is a frog. What happened was that they had dissected the frog. Its anterior had been separated, and only the posterior part of the frog was present, right? And some charred spatulas actually touched the leg of the frog while they were, skinning this frog, and they saw that the leg twitched or it jerked, right? So he thought that maybe there is some electrical property that exists in the cells of the leg of the frog, because of which this twitch or jerk occurred, right? Then he tried to actually stimulate them, and he saw that similar kind of response was observed, right? So he deciphered that there is electrical property in the muscle and the nerve cell that is present. Then these three famous scientists, Johannes Muller, Emil Dubois, Boyce Riemann and Hermann von Helmholtz, they were German physiologists who basically measured the speed of conduction of electrical activity along the axon of nerve cell, right? And they showed that electrical activity of one nerve cell affects activity of adjacent nerve cells in predictable ways, right? So individually also we discussed what exactly they did, right? All in all, they basically developed they found that there is something known as nerve impulse or nerve action potential that is present, right? And they also discovered that the conduction of this nerve impulse is much slower than the electricity conduction, because that is also electrical gradient. This also mostly is an electrical gradient, but it is much slower, and they figured out the reason for that, which was that in terms of electrical gradient, in our wires, in the electrical cables, what happens is the conducting particle is an electron, right? Over here, however, the conducting partner was an ion. It was much bigger. That is why the speed of conduction was much slower, although it was still fast, right? It ranged from zero point 0.1 to 100 milliseconds, 100 meter per seconds, right? Then pharmacology made the first impact on understanding of nervous system and behavior at the end of 19th century, when these three gentlemen, Claude Bernard, Paul Ehrlich, and John Langley, demonstrated that drugs do not just act anywhere on a cell, but rather bind discrete receptors, right? So what importance it had in neuroscience was basically that it proved that the neuron could select which other neuron to interact with, right? So if a neuron has multiple other neurons in its vicinity, right, it will not interact with all of them, right? What it will happen instead is that if it is releasing some chemical to interact with a neuron, only the other neurons that have the receptor for that chemical will interact with it, right? So that is that is what was discovered at this point. Then there were multiple psychologists also, beginning from the 17th century itself, one of whom was Rene Descartes. He was also a mathematician, right? Uh, what he said was he was of a dualistic view that brain and mind are two separate entities, but he also said that they interact with one another, right? But what he also said was that the point of interaction, he gave a visible sorry, he gave a a tangible point of interaction, which was the pineal gland. And why he gave this particular point of interaction, I have explained in the previous slide, because it was in the center of the brain. At that time, it was thought that it was only one single gland, all other parts were bilaterally symmetrical. This wasn't, and so on, right? Then Baruch Spinoza developed a unified view of the mind and body, like we know of as of now also. And there were multiple other scientists who were split between these two groups, empiricists or idealists, right? Empiricists believe that brain is initially like a blank slate, and later it is filled on with sensory experiences, while idealists basically believe that perception of world is predetermined by inherent features of our mind or brain, right? It is somewhere in between these. So we will study that much later when we study the functional organization of the brain and how it is impacted, how development impacts the brain, how what features are post like post natal, what is the development that is happening, right? That we will discuss. So then, uh, it was Charles Darwin who, in the mid 19th century, basically proposed that to study brain, you don't always have to look at humans, right? Before this, everything was very crude. People generally used to only study the humans to look at how the brain is functioning. He basically advanced the idea that animals could serve as models of human behavior, right? Also, because of these propositions, study of evolution gave rise to ethology, which is investigation of behavior of animals in their natural setting, and to experimental psychology, which was study of human and animal behavior under controlled conditions, right? So this fellow was Charles Darwin. Then came Sigmund Freud, who introduced psychoanalysis, right, which was another field which basically framed enormous problems that confront us in understanding the human mind. Then what is the seat of knowledge? How does the brain respond to certain experiences? Where do memories go? How are memories formed? Things like that, right? Then then Francis Joseph Gall. Gall was of key importance because he was a neuroanatomist who proposed two radical ideas. One, that brain is the organ of the mind and all mental functions emanate from the brain, right? So he rejected that both of these are separate entities. Second, he said that the cerebral cortex did not function as a single organ but contained within it many organs, and particular regions of cerebral cortex control specific function. To some extent, he was correct, but he did another thing to prove this point, which was not very smart, right? It was not very scientifically backed up. So he enumerated 27 distinct regions of or organs of the cerebral cortex, like this, in which he basically assigned intellectual processes to certain areas of the brain, right? So we have gone through this in detail, what he exactly did, right? So his theory was prescient, and his experimental approach was extremely naive, right? Why? Because he was influenced by physiognomy, right? So I told you guys that what inspired him was that during his school days, what used to happen was that his fellow classmates, some of them who were, very good at complex problems, at mathematics, in memorizing things, he just observed a pattern that they have a huge forebrain or something like that, right? And because of which he thought that this forebrain region is the part where, these memory, comprehension, all these, logic, reasoning, etc., they reside, right? Similarly, what happened was once became a neuroanatomist, he was assigned to a mental asylum. Over there, he used to study patients who would have some fixative problem, right? As in, they would fixate on either being very worried about something, constantly worried about something, or constantly aggressive, or constantly, um uh, 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 daydreaming, something like that, right? Or who would be very paranoid, or people who would be very spiritual, right? Extremely spiritual. They are seeing the deities everywhere, something like that, right? So, based on those characterizations and observations, he basically made this kind of a map, right? And what he used to do was he used to sit a person down, used to go through the skull of the person, right? And based on the morphology of the skull, right? He used to say that, okay, this person is like this person is like this, right? So it is a pseudoscience. It is not really true that based on the surface of the skull, you can't tell what is inside the brain, right? So he assumed that size of an area of brain was related to mental faculty represented in that area. These are the faculties that he uh, basically suggested. So, this kind of science was termed as phrenology at that time, right? It is a pseudoscience now, so it is not really true. These ideas were subjected to experimental analysis by Pierre Florence, right? What he did was he took animals and he basically ablated brains of animals, and he checked their response for the mental faculties that had been pointed out by Gall. And he said that that did not hold true, right? But what concept he formed was also not true, that he proposed that, uh, uh, one second, he proposed that injury to any one area of the cerebral hemisphere should therefore affect all higher functions equally. So he proposed a holistic view of the mind, right? That the whole mind is involved in every problem, right? That is also not exactly true, right? So this holistic view was challenged by many scientists. Broca, we will study his, findings. Carl Wernick's findings, we will study today, and Hewling Jackson. Right. So we basically studied a little about Jackson's finding, and also uh, Charles Sherrington, who is this fellow over here, who basically championed the view of brain function called cellular connectionism, right? Uh, we will go through Wernick's work in detail, so that is why I'm not mentioning it over here, right? This is essentially what we did in the previous session, right? So, and we did it in a lot of detail, right? We did not just skim through it like we are doing today. We went on every slide. We discussed every slide. We discussed the rationale behind the scientists thinking in the way that they were thinking, right? So we have gone through that in immense detail, and that recording will be available to you. If not through ISSS, I can upload it on my end, right? If that is not available over there, then yeah, you will not miss out on anything, right? So we are picking up on where we left off, right? So this lecture, I was supposed to pick up components of the nervous system, But if you would have gone through the flyer, there are two essential, there are two kind of similar modules that I have given, right? One is the component of nervous system, and the upcoming one is the gross organization of the brain and the structure of the neuron, right? So over here, anyway, components of the nervous system is not a very huge unit, and I had kept it following that origin of neuroscience because I wanted that origin to go smoothly, right? So you get an idea about where we had started and where we are currently, right? So that part we will discuss first. We will finish it off, and then this part will take like half an hour maybe, and we can discuss this also then, right? So yeah. So continuing with the historical perspective, what we said was, up until now, there were multiple. So I'm not going to give another recap, right? We have just went through a recap, right? And over there also, we saw that there was a mention of Broca and Wegener, right? So over here, we are starting off with who was Broca, what did he study, and what did he contribute to our understanding of the brain, right? And his contribution was so important and phenomenal that there is an area in the brain which is named after him. It is known as the Broca's area, right? So let us go through what he did. So, although phrenology was fatally flawed, we understood what is phrenology. We also understood how it was a flawed science and how it became a pseudoscience because of that. Right? So, the basic idea of different parts of the brain serving different function paved the way for future developments in 19th century. Right? So, phrenology was flawed. Uh, f- uh, Florence tried to prove that wrong, right? He did prove it wrong, but he also gave an explanation that was also fatally flawed, right? He said, so Goyle said that different parts of the brain perform different functions. They are responsible for different functions, right? And he, because of his, functional analysis of that, it was not accepted, right? And then Pierre Florence said that all parts of the brain are responsible for all functions, right? That is also not true. So the first part is partly true, but the second statement is completely false, right? There will be multiple parts of the brain that are involved in one common function, but not all of the brain is involved in all of the functions that we perform, right? So music is actually listening to music is one of the rare functions in which at times the entire brain gets involved, right? That is also at times, not always, right? So yeah. So Broca in 1861 reported two brain damaged patients, right? He presented the reports of two brain damaged people. So let us go through what he did. He documented two cases in which acquired brain damage had impaired the ability to speak but left other aspects of cognition relatively intact. Right? So, what is acquired brain damage? That basically a kind of brain damage that is not present since birth, right? So maybe there was an accident, maybe there was some kind of a stroke attack, something due to which there was a brain damage to a particular area, such that the patients that had that particular area of the brain defective, what happened, they lost the ability to speak. They knew how to speak because since their childhood they had learned the language, they were speaking, but due to that particular accident and damage to the brain area, they lost the ability to speak, right? But all other cognition was still flawless, right? They still knew how to walk, they still knew how to drive, they still knew how to paint, they still recognized people, they still had memory of everything, but they lost their ability to speak, right? So he concluded that language could be localized to a particular region of the brain. Now, that is a good observation, right? That is a similar kind of observation we could also have, right? But we did not exist at that time. We did not have as advanced technology as is available right now. So this was a big observation at that time, right? That he could, based on just two cases, based on studying two cases, his observation skills could have been flawless. That he could come to that conclusion, right? So in 1861, basically he described a patient, right? His name was Lipon, who was who as a result of a stroke could not speak, right? We have gone through this, although he could understand language perfectly well, right? So if you showed him something, right, for instance, he couldn't speak that what he wants to eat, but if you write it, and he couldn't write also what he wants to eat, right? But if you show him uh, uh, in writing that, do you want a banana, question mark, maybe he can respond to that. Is this apple fine? He might start eating the apple, or he might throw away the apple, right? So they knew that the language, he could understand it, but he couldn't speak, right? And why? Because of stroke, that particular brain area was defective. So this person did not have any motor deficits of the tongue, mouth, or vocal cords that would affect his ability to speak, right? So all these things are testable, right? So if there is a motor deficit in the tongue or the mouth, there will be difficulty in eating, swallowing, slurring words, talking, etc. Also, and this person could actually slur out words sometimes. So that is why they knew that the vocal cords were also not defective, right? That will come later. Sorry. Yeah. So he could utter isolated words, whistle, and sing a melody sometimes, right, without difficulty. That is why they knew that the vocal cords were also fine. But he could not speak grammatically or create complete sentences, right? Maybe randomly he will say a word, or randomly he will whistle, right? But he cannot create complete sentences, and he could not express his ideas in writing also, right? So neither could he speak nor he could write sentences, right? Maybe he could just slur out that water, right? That would tell somebody that, yeah, he wants water. But sometimes maybe he would slur out the wrong word, right? And people have to figure out what does this person want, right? So he would think that, like, in his mind it would go on that he's thinking about water, but what he's saying is probably a pen or notepad, right? So it would be difficult for him to convey what he means, right? So if you have basically come across stroke patients, it happens, right? So I have seen it in person also, that they are trying to communicate or convey something, but due to certain area getting affected, they cannot, right? They feel like they are saying the entire statement, but they are not, right? They feel that they are writing the whatever they are thinking, but they just scribble, right? There is no words that are coming out. So that happens. So, this patient eventually he lost his life, and the post-mortem examination of his brain basically showed a lesion in the posterior region of frontal lobe, right? So we have still not studied how the gross organization of the brain is. So all you need to remember is that there was what is posterior? Anterior is the top of the head, posterior is the bottom of the head, right? Or sorry, uh, anterior is actually the front of the head and posterior is the back of the head, right? So that is dorsoventral. That is not anterior posterior. Anterior posterior is forehead and the back of the head. That is back of the head is posterior. So posterior region of the frontal lobe. Where is the frontal lobe? We will study soon in the next session, right? And now this area is actually known as the Broca's area, right? So this area that he identified, it is named after him, Broca, right? Then came another, famous scientist. His name is Carl Wernick, right? We talked about him also last time. So who was Carl Wernick? He was another scientist. He basically came a little after this Broca, and he took the next step in 1876 when he published a paper which was titled The Symptom Complex of Aphasia. What is aphasia? The inability to speak, right? A psychological study on anatomical basis, right? So he did a psychoanatomy study, right? So what he did, he studied the anatomy of the brain, and he also looked at the behavior of the patient. It was similar to what was done previously by Broca, right? In this particular paper, he described another type of aphasia, a failure of comprehension rather than speech. So that was also aphasia that Broca's patient was suffering, right? So it was inability to create sentences, inability to sometimes speak words or to write words or to form, to form sentences. I already said that. Yeah. Over here, there another kind of aphasia was there wherein the person can speak fine, right? So he can speak entire sentences also, but he cannot comprehend the speech, right? He cannot understand what the other person is speaking. So if I'm saying something, right, person will not be able to understand what I'm saying, right? A receptive as opposed to expressive malfunction, right? So he can express what he is feeling, but he cannot understand what I am saying, right? So it is a receptive malfunction. Whereas Broca's patient could understand language but not speak, Wernick's patient could form words but not understand the language, right? So it was the exact opposite of what was happening to Broca's patient. But in both the cases, they were basically diagnosed with aphasia, right? So inability to speak or rather communicate, because in this case the patient could speak, right? He could not communicate because lack of perception of what other person spoke. So the locus of this new type of aphasia was different from the one which was described by Broca, right? So that was where it was a posterior part of frontal lobe of the brain. Over here, it is a posterior part of the cortex, right? So when Broca studied that lesion in two patients, it was always coming towards that frontal lobe posterior end. When Wernick, however, studied his patient who had a slightly different kind of aphasia, he basically localized that particular lesion to posterior part of the cortex, which is the part where temporal lobe, which is also present towards the posterior backside of our brain, meets the parietal and occipital lobes, right? So this was the area that Wernick basically found. It is actually now known as Wernick's area. So subsequent studies argued that the language itself was not a single entity, right? So that is why there are multiple areas dedicated to language, because it is not a single entity. There are multiple, subdivisions within language itself, right? It is subdivided into speech recognition, speech production, and conceptual knowledge, right? It was motivated by the observation that brain damage can lead either to poor speech comprehension and good production, or good speech comprehension and poor production, right? So either somebody could comprehend what you are speaking and not speak, or they could not comprehend what you are speaking and speak, right? So this is a representation of the Broca's area that is present over here and Wernick's area that is present over here, both towards the posterior side. This in the posterior frontal lobe, and this in the posterior of the cortex, right? Okay. Then we move on to Corbinian Brodmann, right? So he was another scientist who has basically divided brain in a certain aspect, right? We are very soon going to understand how he has basically gone through that. Just give me a moment. Yeah. So this fellow, what he did was, uh, just a moment, let me check my battery. I'll turn on the charger. Yeah. He was also an anatomist and a German, and he led a new school of thought that cortical localization at the beginning of the 20th century, right? So, what he did was he distinguished functional areas of the cortex based on shapes of cells and variations in their layered arrangement, right? So, I don't expect most of you to still have gone through how the brain looks like, how the cell bodies in brain look like, right? So but the arrangement is actually very different, right? And soon we are also going to understand about the structure of neurons, how there are different kinds of neurons. So these different kinds of neurons actually exist in different areas of the brain, right? And their arrangement in these different areas is also very different, right? That is why we can actually identify multiple brain areas, right? So cerebellum like has a very stark different arrangement. Hippocampus has a very distinctive arrangement of these neurons. Uh, and there are multiple brain areas that we can identify solely on the basis of how the neurons are arranged over there or how multiple cell bodies are arranged over there, right? So what Brodmann basically did was once he did the staining of the brain, he basically identified that different kinds of cells were present in different kinds of regions in the brain, right? Based on this concept, he did a cytoarchitectonic method, right? That is what he applied, a cytoarchitectonic method. So cyto means cell, architectonic has to do with architecture, right? So based on the architecture of the cells, right, he basically distinguished 52 anatomically and functionally distinct areas in the human cerebral cortex, right? Some of which are shown over here, which are on the surface of the brain. Some of them are within the brain also. So in this view, it cannot be seen, right? So you can see that some are marked with these dots, some are marked with these holes, some are marked with the, a different color dot, crosses, dashes, eyes, etc., triangles, filled triangles, empty triangles, etc., right? So, based on how the cell bodies were arranged or how cells or neurons were arranged in these areas, he divided the cortex into 52 different, parts, right? And he firmly believed that these different parts did different functions and because he said that different cell types are why are they different from one another? Because maybe they need to perform a specialized function, right? Based on that rationale, he did this division and he believed that there are 52 types of anatomically distinct regions that are present in the brain that control distinct functions, right? Then after this Brodmann theory, right, came Lashley, right? So he was very skeptical of the cytoarchitectonics approach to functional mapping of the cortex, right? So he was not very convinced with this approach that Brodmann had taken, right? So what he did instead was he and after him, multiple other psychologists concluded that learning and other higher mental functions have no special locus in the brain and consequently cannot be attributed to specific collections of neurons, right? So, this is talking about higher functions, right? But is it true in all regards? So, it is not entirely true, right? Because listening, speech comprehension, language, etc., is also a higher function. And Broca and Wernick were able to basically, locate the brain areas or the brain loci, which they were, bound to, right? But it would have been possible that those brain areas had a mixture of neurons, right? So it could be that Brodmann was not true, but Lashley was also not entirely true, correct, right? So, these are the kinds of things that people have thought of. The rationale behind their thinking was often, good, but, scientific evidence let one of these thoughts prevail and the other it, proved incorrect, right? So that is why we are studying all these kinds, because these kind of questions might arise in our mind also, right? And they should. So, because that is how science develops, right? Something will come up in your mind, and then you have to work towards either proving it correct or proving it incorrect, right? That is what you do in research also, right? You take a hypothesis based on certain things that you already know or certain literature that already exists, right? You think about something, you formulate a possible answer for that, and then you work towards proving whether this is the correct answer, right? So then you need scientific evidence to back up your answer also. So that is what research is, right? That is what I'm doing with my hypothesis also. Correct? Okay. So Lashley basically reformulated the aggregate field view by further minimizing the role of individual neurons, right? Sorry, specific neuronal connections and even specific brain regions in the production of specific behavior, right? So, as I said, he did not believe that any of these things held true, right? He believed in, again, a more, holistic kind of view. Not exactly holistic, but he did not believe that specific neurons performed specific connections or functions, right? So, according to Lashley's theory of mass action, right? So, as the name suggests, again, it encompasses a lot of or whole of the brain. It is the full mass of the brain, not its regional components, that is crucial to function, right? So, kind of like holistic view, right? So, we know that holistic view is not true as of now, right? Then later on came Wilder Penfield, right? So he is a remarkable scientist. He is one of the people uh, whose work has basically given us the most knowledge I can think of about which part of the brain is doing what function, or possibly which part of the brain is involved in which function, right? So he was an American Canadian uh, neurosurgeon, right? He, between 1928 and 1947, he and his colleagues carried out series of remarkable experiments on over 400 living human brains, right? So, as I had given a brief introduction about Wilder Penfield in the previous session also, uh, like last time when we met on Friday, I told about him that he was basically an epileptic neurosurgeon, right? So he used to deal with people who had epilepsy. Epilepsy is basically a very hyperactive condition of the brain, right? Where the brain activity is um unnaturally hyper, right? So the brain waves are very fast, and because of that there is electrical imbalance in the brain, due to which there are seizures that occur, right? Uncontrollable movements that occur. There is the normal function of the brain is lost when a person is experiencing such epileptic activities. So that is the condition that he used to treat, right? He was a neurosurgeon for epileptic patients, right? So he had a lot of patients that used to come in. Now, epilepsy, now with development of science, there are more and more techniques that are coming up for treatment of epilepsy, right? Few of which are drug targeted, but they are effective only in handful of population. Few of them include electrical therapy of sorts, wherein small bursts of electricity are given in your brain, which will neutralize the basically erratic electrical activity that is going on in your brain, so that you will not get seizures, right? So those kinds of studies are also coming up and also been clinically pursued, right? But back in this period, when Wilder Penfield was actually, practicing, what used to happen was that if a person gets epilepsy, and if that epilepsy is a burden on the person's livelihood, right? Somebody could get seizures very frequently, like multiple times a day also, right? So it will be very hard for that person to, pursue a normal life. So what practice was followed at that time was that there was a open brain surgery that used to be done in these patients, wherein the focal point or the origin point for this epilepsy used to be identified through with the help of electrodes that were implanted. And then if it is not a very life sustaining part of the brain, that is it is not controlling the function related to heart beating, respiration, digestion, etc., right? If it is just associated with some secondary functions such as memory, cognition, walking, or short-term memory consolidation, or speaking, etc. What used to be done was that because the person is still going to be alive, if that portion is removed, it is not going to hamper with the uh, uh, life of the person anymore. That part used to be removed. It was known as lobotomy, right? So that part of the brain used to be removed, so that the epileptic region of the brain is now gone. So now the person is not having any seizures. Although they might have lost the ability to walk, although they might have lost the ability to speak or to comprehend speech, etc., right? But it was like a cost to benefit ratio, right? So, in order for the person to live seizure free, that is a cost that they had to pay, right? So, at that time, it was a very common practice. It is a common practice even now, right? Sometimes, uh, because it is a very difficult condition to sustain with seizure, right? And it is fatal also, because you don't know when you are going to get a seizure. You might be driving and you get a seizure. It will be harmful for you, the person in the car with you, the people on the road next to you, right? So it is a fatal condition altogether. So depending on where or which circumstance you are in, right? Because of which this was a crude approach, but it was still widely used, right? And it is still widely used. So, what he did was, the patients in question were undergoing brain surgery for epilepsy. I explained what epilepsy is, and I explained how this brain surgery used to go. Right now, let us come to the details of it. Right. So, what his experiments used to be like was to identify and spare regions of the brain involved in movement and sensation. Penfield electrically stimulated regions of cortex while the patient was still conscious, right? So, in a brain surgery, actually, even if you have seen accounts right now, the patient is actually conscious during a brain surgery, right? That is how a brain surgeon, sorry, or a neurosurgeon gets to know that they are, at the correct region in a brain, right? Because we still don't have the understanding of the entire brain, right? So if they just poke a little bit uh, off than what is expected of them, they might, critically hurt that person in a way that the person is maybe gasping for breath, or the heart function goes, array. Uh, like that, right? So the person is always conscious during a brain surgery. Obviously the person is under anesthesia also of some kind, because the drilling, the brain itself doesn't have any pain receptors, right? So the brain cannot feel any pain, but the skull has pain receptors in them, right? So the drilling part of the skull, that is painful. During that, the patient is either put to sleep, or put down, put under, or they are given anesthesia of a sort that it will keep them awake, but they will not feel the pain, right? So, Penfield basically used to stimulate regions of the cortex while the patient was still conscious, right? So, once the brain is opened up, he used to stimulate different regions, right? Because he had electrodes that he obviously needed also to locate the focal center of the epilepsy. While doing that, he used to make notes about all the areas of the brain that he used to encounter, right? Because it is not all with techniques now, with the kind of imaging that is available now, with the kind of other techniques that are available now, it is possible that you have correct idea about which part of the brain you need to focus on, and in one or two go you can just locate that area and just lobotomize that area. But at that time the imaging was not that advanced. You needed to surf around the brain to find the point of epilepsy, right? So when he was surfing around, he used to encounter patients basically saying that they have some kind of sensations, right? We will read about them also. So that is why he started making notes of these sensations, right? Based on which area he was at. Or poking at that time, he started making records of sensations of the patient. So the procedure was not painful, but patients did report some fascinating experiences. We can read about these experiences, right? So, for instance, when he used to stimulate the occipital lobe, right? One patient reported that a star came down towards my lobe, right? So it could be that he's either seeing a shooting star, or he's recollecting some memory, or it could be that he's basically dreaming of something, or he is creative, like his creative mindset was over there and he's thinking of something that could never happen, something like that, right? So, upon stimulating a region near the central sulcus, how another patient commented, those fingers and my thumb gave a jump. Right. So, although the surgery was going on over here, nobody was touching the fingers and the thumb and the toes, right? But the patient felt like something happened over there. Some sensation happened in the fingers, right? When he was near the central sulcus region. After temporal lobe stimulation, another patient claimed, I heard music again. It is like the radio, right? So she would have heard some music while coming maybe, or a few days ago maybe, and she was able to recall the tune she heard and was absolutely convinced that there must have been a radio in the operating theater, right? She did not think that it was her mind who was thinking that she heard something. She was definitely sure that she heard something, right? And it was playing in the operating theater itself, right? Just because some part of the temporal lobe had been poked, and it probably recollected some memory pertaining to that particular music experience, right? So, of course, the patients had no idea when the electrical stimulation was being applied, so they couldn't feel it or see it, right? As far as they are concerned, an electrical stimulation applied to the brain felt pretty much like a mental or cognitive event, right? So they actually felt like they were doing it, right? It did not feel like the brain was just, being stimulated in a certain way that made them think that they were doing it. They actually felt that they were doing it, because all these, sense of self, sense of what is true, sense of what is incorrect is coming via our brain only, right? Via the functioning of our brain itself, right? So that is why, because their brain was functioning in a certain way, they actually believed that experience, and they actually felt that they were, experiencing a mental or cognitive event, right? Here I have presented a timeline for the development of methods and findings that were relevant to cognitive neuroscience, starting from phrenology to the present day, right? So we are slowly shifting our focus from understanding the brain to understanding physiology to understanding psychology to understanding to where phrenology came into picture to that neuroanatomy and localizing brain areas to finally understanding cognition, right? So this particular timeline, we are starting from when phrenology was put forward, right? So it was something somewhere in the early 1800s, right? That phrenologists basically put forward their localizationist manifesto, right? We have understood what the phrenology was, and we know what they meant, what is meant by their localizationist manifesto, right? Then after this, in the mid 1800s, Purkinje, right? So there is actually one kind of neuron that is named after Purkinje. It is known as the Purkinje fibers, right? So that was the first kind of nerve cell that was described, right? It was done by Purkinje in 1837. Later, Broca in 1861 published papers on language localization. Right, we have looked at a lot of studies by Broca also. Then, in shortly after that, Fritz and Hitzig in 1870 they applied electrical currents to the dog cortex and they caused movement in these dogs. Right. Later in 1929, EEG was developed as a research tool, electroencephalography, that studies the brain waves. Right. Then Hodgkin and Huxley in 1939 discovered action potential and they enabled single cell recording, right? So we will read about Hodgkin and Huxley in detail also when we read about signaling in the brain, right? So they did this experiment in basically squid axon, which has a very huge axon, right? So we'll come to it, but action potential was basically discovered at this time. Then influential publications by Broadbent, Chomsky, Miller, and others basically led to emergence of cognitive psychology. Right. Later techniques like CT and MRI were developed. Right. These are both imaging techniques that are very, very applicable even in today's viewpoint. Right. Then in vivo blood flow measurement in humans was developed, enabling PET in 1979. Then there was the first study of TMS that was reported by Barker in 1985 and later 1990s, BOLT, which is also blood oxygen dependent response basically, was reported enabling fMRI or functional MRI development by Ogawa et al. In 1990. Right. So this is eventually how techniques developed, how our understanding developed, right? So conceptual understanding we have gone through in detail, right? We have understood that who were the famous neuroscientists of their time, right? What were the theories they put forward? What was correct in those theories? What was incorrect? Even if the entire theory was incorrect, how did it lay groundwork to basically come to a correct theory, right? All this we have studied, right? Now the logic or the rationale behind this slide is to basically promote uh, these various techniques that were developed to further the study of the brain, right? So the birth of cognitive neuroscience, right? So as I told you, one of the books that I am referring to for this course is this book, The Student's Guide to Cognitive Neuroscience. It is written by Professor Jamie Ward, who is a professor at University of Sussex in UK, right? So while Candle, basically the other book. that is principle of neural science, which is Candle et al. While that basically deals with um, more of cellular and molecular and signaling mechanisms. This particular book deals more with cognitive neuroscience, right? So we are going to have a mix of the two in all our lectures so that you basically understand both perspectives equally well, right? So when we speak of cognitive neuroscience, cognitive has to do with something that is related to higher order thinking, right? Higher order thinking or processing is known as cognition. So, cognitive neuroscience was largely advances in imaging it was largely advances in imaging technology that provided the driving force for modern day cognitive neuroscience, right? So, technology such as EEG, which imaged the electrical pattern of the brain waves, right? Or the brain by itself through CT, MRI, fMRI, PET, BOLT, etc., right? Also, it is important to note that technological advances in imaging not only led to development of functional imaging but also enabled brain lesions to be described precisely in ways that were never possible before, right? So, how these were the different methods through which it occurred, right? A lot of these are non-invasive methods, but some of them are invasive in nature also. That is invasive meaning anything that will uh, cause even a minor distress to your body. So even if you are injecting something, it is invasive. If you are not injecting anything, if you are just imaging without touching the body, without harming the body in any even a minute sense, it is known as non-invasive, right? So there were multiple methods such as EEG, ERP, single cell recordings, TMS, TDCS, MEG, PET, fMRI, etc. Most of which were based on recording. Some of them involved stimulation of the actual brain area, that it was first stimulated and then it was recorded, right? As I said, most of them were non-invasive, but a few of them were invasive. A single cell and all, for example, was very invasive, and it can only be done in vitro settings or in non-human animals, right? So in humans, it can't even be done, the single cell recordings, right? Then the brain properties, which they were basically getting a reading of. So EEG, single cell recordings, they were basically recording the electrical activity of the brain. Even CMAP, etc., right? They would also record the electrical property of the brain. TMS would record the electromagnetic property of the brain. TDCS would record the electrical activity of the brain. MEG, as the name suggests, is magnetic encephalography, right? So it will record the magnetic properties of the brain. Wherever electrical properties exist, magnetic properties will also by default exist, right? And PET fMRI, they were basically recording the blood flow or the hemodynamic properties. So they used to tell about which brain areas are active based on where the blood flow is more or where the blood glucose that is there, and that is basically labeled, radio labeled, so we can follow that glucose where that glucose uptake is more, right? So they would use the hemodynamic brain property to basically tell what is going on in the brain areas, right? So the distinction between recording methods and stimulation methods is crucial in cognitive neuroscience. While direct electrical stimulation of brain in humans is now rarely carried out, right? Earlier, the methods were much crude, right? So now the methods are much more precise, right? People don't go for very crude methods anymore, right? Only if lobotomy is inevitable will that happen, right? Otherwise, people will rectify it in some way. So the modern day equivalent of these studies uses stimulation across skull rather than directly to the brain, right? So rather than opening up the brain, creating a cranial window, going inside, poking an electrode in the brain itself, what people do now is they will shave off the head from wherever they want, to, stimulate your brain. And transcranially, that is from above the skull itself, they will stimulate the brain, whether it is ultrasound based stimulation, or it is magnetic property based stimulation, or electricity based stimulation, that is varying, right? So yeah, we will shortly study or look at the methods of cognitive neuroscience, although I have not laid much emphasis on this particular portion. And why I have not laid much emphasis on this portion is because I had seen that there were a couple of courses that were talking only about what are the methods that are used to basically image the brain or look at the brain, etc. So I did not want there to be much of an overlap. But still, if you feel like you want me to explain this, you can let me know. So I will include it in my upcoming slides, right? So we will briefly go over this topic, but I have not, I like that is not what I planned, at least, that I will not go into the principle of each and every kind of, method. But again, if you want me to, I can include that. So you can let me know, right? Just give me a short break. I'll just resume. Yeah. So, as I was talking about the methods that we use to study the brain, and over here they are listed as the methods of cognitive neuroscience, because we have picked up from this particular book, right? We talk about some properties that they should have, right? So, these properties are what is the resolution of the brain that we can see, right? So, for instance, when you are picking out a phone, right? You will check certain features for the phone, right? Some features will be like, is it handy? Because now most of the phones that are coming, the mid-range ones or the low-range ones at least, they are very bulky phones, right? So they will all be like 6.3 inch to 6.6, 6.7 inches, right? So earlier that used to be really fascinating for people, but now, but it is less practical, right? It will not fit in everywhere. So some people will want bigger phones that, okay, anyway, I carry a purse around, or anyway, like for guys, for instance, they have bigger pockets. So it is okay for them that, yeah, I will anyway carry a purse and I don't keep my phone in my pocket. So, I can go for a bigger screen phone. But for some people, they would want that, I want a more compact phone because it doesn't really fit that well into my hand, and I keep dropping it because of that. Right, so that is one instance. The other instance that you might look at would be the battery life, right? So, somebody who is at a desk job, for example, or for me, I'm in a lab, right? So if I was in a dry lab, I would have work, usually desk work, right? So even if the battery life of my phone is not very nice, I can do with it, right? Because I can just keep it on charging whenever I want because I have to work on the desk most of the day. So I can afford that, right? I can afford the battery to be not that good. But somebody, for instance, who is in a field job, sales, for instance, right? So that person is going from door to door at times selling stuff, right? So they don't get access to charging or a power bank all the time, right? So that person will require a phone that once the person charges it in the morning, it will run the entire day long, right? So there will be a different criteria of filtering what I want in a phone, right? Similarly, somebody would be a fanatic who loves to click pictures, right? So somebody who loves to click their own pictures or who is a selfie lover, right, will want a good front camera. The camera should be in the center, not on the side, something like that. These criteria might be there. Somebody who is a nature lover, nature photographer of sort, right? They will want a fabulous back camera. They will probably want even a point 0.6 x lens wherein they can have a wide angle view also, right? So there will be different method like based on the need. uh, the filtering criteria will differ, right? So similarly, there are different kinds of criteria or characteristics of every technique also that exist, right? So what will be a perfectly ideal technique or method uh, for assessing the brain? It Okay, first let us go through what these things are and then we will discuss about that, right? So when we talk about temporal resolution, for instance, this is one of the criteria, right? So like battery power or like camera resolution, right? So for instance, my camera has a 50 megapixel resolution and another camera has a 25 megapixel resolution, right? What I understand, like if I don't go into details of it, like what is the processing power of that particular phone, etc. If I don't go into those details on a grosser level, a 50 megapixel camera will be better, right? Uh, Although I'm not saying that is always the case, right? But just in layman's term, if we are speaking, that will be better, right? So similarly, temporal resolution is basically a resolution pertaining to time, right? So how accurate it is with respect to time, right? So what is written is it refers to the accuracy with which one can measure when an event is occurring, right? So, for instance, we are saying that neurons are firing like in milliseconds, right? In fractions of seconds, neurons are firing. So, do we have techniques that can basically capture when these neurons are firing, right? So, they should have a high temporal resolution, milliseconds of resolution they should have. They should not be in seconds or minutes. They should have millisecond worth of resolution, right? For us to study the brain property, this particular brain property that is firing of neurons, right? Or synaptic transmission. Okay. So that is what temporal resolution means. The effect of brain damage are permanent, right? For example, if we are studying brain damage, the effects are permanent. And so that does not have any temporal resolution as such, right? Because it is kind of like an endpoint study, right? So in an epileptic patient, for instance, you can you can do a study or you can use a technique that has a good temporal resolution so that you can understand when the brain is firing, when the synaptic transmission is taking place, etc. But if you are doing similar kind of a study with a good temporal resolution on a stroke patient, what will you get, right? In case of a stroke has already occurred, a area of the brain has already been damaged, right? Now you want to look at where the damage is localized, or what part it is affecting the most, right? You don't need, time is not a constraint over there, right? It has already been done. It is an endpoint situation, right? So nothing is happening in real time now over there. right? So even if it doesn't have a millisecond or fraction of second of temporal resolution, it is fine, right? So that is what the statement is saying, that effects of brain damage are permanent. So it has no temporal resolution as such, right? So because it has already been done. So methods such as EEG, MEG, TMS, and single cell recording have millisecond resolution, right? So that is what I was saying, that uh, better temporal resolution, meaning millisecond temporal resolution, right? Not minutes or seconds of resolution. It means that it should capture changes that are going on even in milliseconds or fractions of seconds, right? fMRI has temporal resolutions of several seconds that reflect slower hemodynamic response, right? So, for instance, electrical activity, neuronal synaptic transmission, action potential, all these activities are quicker, right? They are electrical signal based. But blood flow, right? Blood flow to a certain region is a much slower process, right? It happens over seconds. It doesn't happen over milliseconds. It happens over seconds. So that has a temporal resolution of several seconds. Even if something does, like for instance, if I am not that fond of clicking pictures, and that 25 megapixel camera phone also has a fabulous battery, and I'm a salesperson, right? I would probably go for that, right? That is much better, and it is solving my objective much better, right? So that is what is written in terms of fMRI. Then there is another kind of resolution that exists. Temporal meaning pertaining to time. The other is spatial, that is pertaining to space or pertaining to where it is located, right? So it refers to accuracy with which one can measure where an event is occurring, right? So what does that mean? That it should have probably, it should point out to the exact location in the brain, in microns. There is meter, then there is centimeter, millimeter, then there is like 1 raised to power 10 raised to power minus 6 of meter is a micron. Then there is a nanometer, 10 raised to power minus 9, femtometer, 10 raised to power minus 12, right? So that kind of a resolution, if it is possible via some methods that are present for assessing the brain, right? So that kind of accuracy in microns or in picometers, etc., that is what we are looking for. So lesion and functional imaging methods have comparable resolution at the millimeter level, whereas single cell recordings have a spatial resolution at the level of a neuron, right? But again, we will study that that single cell recordings are not very practical because we cannot really do them in humans also, right? It is difficult to do. It is doable. Patch clamp is not doable. Single cell recording is doable, but it is difficult and it is very invasive in that sense, right? So over here, for a good spatial resolution, where will we require it? For instance, again, a person had a stroke, right? Now the person has already had a stroke. Now we want to treat the stroke, right? Treat the area of the brain where the stroke had occurred. So we want to know the exact brain area where the stroke had occurred, right? We don't want to, miss that area, so that whatever we are delivering or whatever, ultrasound wave that we are giving or something treatment that we are giving is targeted to that area and it doesn't have any off-target effects, right? So for that, we need a good spatial resolution, right? Apart from this, yeah, so this is a map which basically on the y-axis, if you see, these are from top to bottom. They are decreasingly, uh, increasing spatial resolution from top to bottom, right? So brain is the least spatial resolution. It is the entire brain. Then comes the map of the brain, column layer. Then neuron is a single cell. So a single cell is assessed, meaning a lot more spatial resolution than was there at the brain level. Dendrite is a part of the neuron also. So that is even further spatial resolution, and synapse is a tiny gap or junction that exists between two neurons. So that is even further in this spatial resolution, right? And in the x-axis, from left to right is decreasing temporal resolution, right? So day is the minimum temporal resolution and millisecond is the maximum temporal resolution, right? So I hope you are understanding or getting what this maximum temporal resolution, minimum spatial resolution, etc., what it means, right? So these are certain techniques which have been plotted with respect to what is their what is their spatial resolution and what is their temporal resolution, right? So, for instance, MEG, ERP, TMS, functional MRI. First let us look at MEG, ERP, and TMS, right? So, all of them have less spatial resolution, but high temporal resolution, right? So, they are basically recording the electrical activity and all of the brain in order of milliseconds and seconds, but the level at which they are recording is not very localized. It is a whole brain or a whole cortical area, something like that, right? Then functional MRI, because now it is recording the hemodynamic response, which is anyway slower than the electrical response. PET, which is also recognizing the hemodynamic response. Now, these are also at a lower temporal scale, and because they are also doing it at a whole brain level, it is at a lower spatial resolution also, right? Similarly, naturally occurring lesions, if a condition already occurs in a patient, right, that is also probably going to be at a low temporal, because the lesions have nothing to do with the time at which the recording is done, and high spatial resolution also, right? Similarly, there is multi-unit recording and single cell recording. So single cell means one cell at a time, multi-unit means a group of cells at one time, right? Not the entire brain, not an entire region, but a group of a few cells at one time is multi-unit, and single cell at one time is single cell recording, right? So you can see that the spatial resolution for them is, these are much better, but and for some of them, even the temporal resolution is much better, right? But now you can also see that these are kind of color coded, right? So I don't think that they are correctly color coded over here, but so we will not go with the color coding. We can individually look at them, right? So a few of these techniques are actually um, very invasive, right? So even if multi-unit recording and single cell recording have the best temporal and spatial resolution out of all of these, they are very rarely done, right? Why are they very rarely done? Because they are highly invasive techniques, right? You will not specially open the brain of a person whom you merely suspect that something is wrong, right? That is not the correct practice also, and that is a very um, naive thing to do also, right? Because there will be much more complications that will exist because of the surgery itself, right? So that is why these are rarely used. These, on the other hand, are less invasive, right? PET again is a little invasive because a radioactive tracer is being injected into the body to basically follow the glucose uptake by the brain, right? So it is a little invasive, but these are highly invasive, right? fMRI and all are non-invasive because they are just imaging the brain. They are not really injecting anything, or no surgical procedure of any kind is being done, right? No cut at all, no point at all is into the skin. So, yeah, these were the methods of cognitive neuroscience that can be categorized according to the spatial and temporal resolution. Now, along with spatial and temporal resolution, I also told you about another criteria of selection, which is invasiveness, right? So now that we have these three criteria, we can uh, basically think of the fact that which kind of a technique will be a perfect or ideal technique to study the brain. It will be a technique which has low temporal resolution, low spatial resolution, and is less invasive, right? So currently, there is no technique like that which exists, but hopefully, with the advancement of this field, that will also come, right? So, that will be an ideal situation, right? And our knowledge, we might be able to expand it much more than what we know about brain right now, right? Yeah. So, after this, we come to contemporary history, right? That is the modern or very recent history. There are just a few of these scientists that we want to talk about over here for now. Right? Obviously, there are so many people, right? There are people who have discovered specific areas of the brain. There are people who have gotten Nobel Prize, right? Recently, also people who have gotten Nobel Prize who have discovered new kinds of neural cells and the satellite or the space cells in the hippocampus, or they have identified that the hippocampus is basically responsible for our spatial orientation. All these things have been done, right? But pertaining to this discussion, only we are talking about the contemporary history right now. So that is why we are not going into those discoveries as of now, right? That will come more in play when we are discussing the physiology more, right? So, as for contemporary history, I have mentioned these three scientists. Joy Hirsch, she is Joy Hirsch. Then Maria Christina Musso, I couldn't actually get an image for her. And Andrea Moro, who is this person, right? So, in separate studies, Joy Hirsch and both of these they have worked together. Maria Cristina Musso and Andrea Moro have worked together, and Joy Hirsch has worked separately. And together, all together, they came to the same conclusion, but they were studying it at different places and, not aware of that the other person is also doing the same study or similar study. They have basically used fMRI to explore more deeply Wernicke's idea that Broca's area contains the grammatical rules of language, right? So, this was what was proposed, right, by Wernicke, that Broca's area is responsible for grammatical manifestation and perception of the this thing language, and Wernicke's area is actually has to do with understanding or perception of language, right? So Hirsch and her colleagues made the interesting discovery that processing of one's native language and processing of second language occur in distinct regions within Broca's area, right? So Broca's area was responsible for the grammatical consolidation of language, right? And what she and her colleagues discovered was that between different languages, for instance, if I know multiple languages, for me, I know Hindi, English, Punjabi, and a little bit of German, right? And a little bit of Sanskrit, what we had done in school, right? So, because out of this, Hindi, English, and a little bit of Punjabi is my native language. So, they will probably be, localized in one area within the Broca's, one region in the Broca's area, and German Sanskrit, because they are acquired much later, right? Uh, Oh, sorry, the native language for me will be Hindi and Punjabi, right? Even English is my second language. But since that also I've been studying since childhood, right? Almost around the same time that I learned the other two languages also, right? They will still be in the same area, is what they have discovered. But the languages that I have derived as an adult, right? That will be processed in a different area, right? So that is what they have said. If the second language is acquired in adulthood, it is represented in a region separated from that which represents the native language, right? So we will see also, visually also what this means, right? If the second language is acquired early, however, both the native language and the second language are represented in the common region in Broca's area, right? So that is another, phenomenal thing that they have pointed out. So what they mean over here, these are images that are representing the functional magnetic resonance images, that is fMRI images of the brain of bilingual subjects, right? So they know two languages over here. One, on A, the languages are Turkish and English, and B, the languages are English and French, right? So they are bilingual subjects. During a generation of narratives in two different languages, this fMRI was taken, right? So whenever they were asked to speak in or consolidate some sentences in Turkish, it looks like red color over here. And whenever they were asked to speak in English, it looks like yellow color over here. And as you can see, if the yellow and red color overlap, it will look like orange, right? So you can see a great deal of overlap over here, and the center of mass of both these individual languages is also falling in this common area, right? This is an early bilingual, right? So one person who had acquired both these languages in the early part of his life, right, or her life. So, what is a late bilingual? Somebody who had acquired a particular language. Over here, the second language is French. So, he was a native English speaker, but French language was acquired later in adulthood, right? So, this is what it looks like. When the person is asked to speak in English, this red area is what lights up. And when they are asked to speak in French, this yellow area lights up. You can see that the point of overlap is very little, or over here, actually, there is no overlap at all if we look at the pixels by itself, right? So, the center of mass also thus is within the individual languages and separate only. It is not in a common area, right? So that is what was found basically by Joy, right? This is what she and her colleagues had discovered, right? That this is what happens. So they could discover this because of a rationale that they had based on a theory or idea that was put forward by Wernick, and they could test this theory or idea because of the availability of this fabulous tool that is fMRI, right? What does fMRI do? It will basically uh, uh, when we are doing functional magnetic resonance imaging, what will happen is based on what study we want to do, we ask a person to perform a certain tasks while we are imaging their brain, right? And we see that which part of the brain is lighting up. How will that occur? How will that part of the brain light up? Because the uh, oxygen, that is fMRI, is basically assessing the oxygen that is going to these brain areas, right? If more amount of oxygen is going to any particular brain area, they will basically light up as these different colors, right? So, and the basal activity or basal oxygen level will remain as this grayscale color, right? So that is what is the principle of functioning of an fMRI basically, right? So I hope you understand this particular result. Then another contemporary history that I wanted to talk about was with Musso and Muro, right? So this is Andrea Muro, and they, with their colleagues, basically found that region of Broca's area concerned with second language becomes established and increases in activity only when an individual learns a second language that is natural. That is one share that shares the universal grammar, right? So, yeah. This Broca's area is very selective in whether it will accommodate the new language as it is a natural language or not, based on whether the grammar is the same or not, right? If the second language is an artificial language, a language that violates the rules of universal grammar, activity in Broca's area does not increase, right? So that means that for instance you are learning a new language that is not falling into the grammatical rules that were followed by your natural native language, right? So in that case the activity in the Broca's area will not increase, right? So it is not probably being localized in that Broca's area, or that area is not being responsible for a lot of the changes that are going on due to the second language. Thus, the Broca's area must contain some kind of constraints that determine the structure of all natural languages, right? So that means that this Broca's area has kind of like a brain of its own. It can make a decision whether I want to give neurons to this particular language, or I don't want to dedicate any neurons to that particular language, right? Now, this is a series of scan, which is brain scan using PET, positron electron emission tomography, right? So, in this, what is done is an injectable tracer element is basically intravenously injected into a patient or a subject, right? Not necessarily one a diseased person. So it is injected into a subject, and then their brain activity studied based on where we are finding this, because it is a radio tracer, right? So it is radioactively labeled. So when we are imaging the brain, we will be able to trace this radioactive element. And this radioactive element will be present at a basal level throughout the brain. But again, like fMRI, over here also, if a particular brain area is active, more of this particular tracer will be directed to that area. Why? Because this tracer is a glucose tracer, right? And brain needs glucose for its functioning, right? And okay, I will come to that point later, that how much of glucose actually is required by the brain, right? So, based on this principle, we can see that which part of the brain is accessing the most glucose based on what we are asking the subject to do, right? And that is how we are localizing that particular brain area to that particular task, right? So, for instance, if we ask a couple of subjects to look at words, right? This is how the PET scan looks like. So, reading a single word produces a response both in the primary visual cortex and in the visual association zone. So you don't know these terms right now because we have not gone through the brain areas as of now, right? But we will come to that. But you can see that a particular area of the brain is lighting up when subject is looking at the words, right? So if they are giving a paper, given a paper, and 10 words are written over there, they are looking at those words, right? So this is the activity of the brain that is happening. If they are listening to those 10 words, right? So instead of being given a paper with those 10 words, they are made to sit at a place, and then a person speaks those 10 words, right? So this is another part of the brain actually lights up. So this is the area that is probably responsible for that particular perception that is coming through audio signals, right? So hearing a word will activate the temporal cortex and the junction of the temporal parietal cortex, right? So this particular area it will activate. If, for instance, these people have to speak the word, right? So they were given 10 words earlier, or they are asked a question now and they have to speak upon it. And once they start speaking, we are starting recording of their brain, right? So these subjects were basically asked to repeat a word presented through earphones or on a screen. The spoken word activates the supplementary motor area of the medial frontal cortex. Broca's area is activated whether the word is heard or read. Okay, so in either case, even if we are giving the word as an audible signal or visual signal, Broca's area is always activated, right? Both visual auditory pathways converge in Broca's area and the common site for motor articulation of speech. So this is something that has been established now, right? So if we are speaking something, then this is a part of the brain that is lighting up. And if on the other hand, we are thinking of something, right? So multiple parts of the brain are lighting up, right? So when the subjects were asked to respond to the word brain with an appropriate verb, for example, to think, this type of task activates the frontal cortex as well as Broca's and Wernicke's area, right? So frontal cortex is being activated. The place where consolidation of words, grammars is occurring, that is being activated. The place where we are comprehending what has been said to us, the perception of language, that is also being activated. Everything is occurring at once, right? Uh, just a moment. I think I have changed the layout. Anyway, not big of a difference. Yeah. Then these areas play a role in all cognition and abstract representation, right? So that is it. So this is what the brain looks like after PET scan, right? And after they are asked to basically uh, look at, sorry, to perform different tasks, right? So this is a consolidation of what we have read, right? So words and the brain. It is kind of schematic by Linda Robinson, wherein they have marked that this particular area is for language production. It is a Broca's area, and it thinks about words mainly. Reading words is more of a visual challenge, so it is present in the occipital lobe. Hearing words is more of an auditory challenge, and it is present in the Wernicke's area where language comprehension is happening. And saying words, because it is requiring the movement of your lips, of your mouth, of your vocal cords. That is why it is in the motor cortex, right? And it is what is saying the words, right? Yeah. So there was another set of case studies that was done, wherein what they did was that they studied deaf people who have lost their ability to communicate even through sign language after suffering cerebral damage, right? So there were deaf people, they could talk through sign language, but they suffered some cerebral damage. And after that, these deaf people even lost the ability to, communicate via the sign language, right? So what happened over there? This American Sign Language or ASL uses hand gestures rather than sound and is perceived by sight rather than sound, but has the same structural complexity as spoken languages, right? So, what happened was the cerebral damage was there, but it doesn't mean that they were unable to see after that, right? They were still able to see. But what happened was that when we are communicating anything, right? Gestures, for example, if I say this is not a visual signal only, right? You can see me doing this, but your Vernix area is also probably active because you are comprehending that what does this thumbs up mean, right? Does it mean that I have done a good job? Does it mean that this part is correct, right? Or if I do a thumb down, does it mean that this whatever I'm saying is untrue or whatever you are doing is not correct, something like that, right? So although the stimulus is visual, but the comprehension of the stimulus is similar to comprehension of spoken languages, right? So it is perceived by sight rather than sound, but has same structural complexity as spoken languages, right? Thus, signing is also localized to the left hemisphere, right? So the deaf people can become aphasic or they can lose their ability to speak or to communicate for sign language as a result of lesions in the left hemisphere. Right, but not as a result of lesions in the right hemisphere, right? Because right hemisphere is not responsible for those kind of activities. It's the left hemisphere that is responsible for this activity, right? So these observations basically illustrated three major points. One was that cognitive processing for the languages occurs in left hemisphere and is independent of pathways that process the sensory and motor modalities used in language, right? Whether it is audio, visual, or it is motor because it is pertaining to speech, that is it is independent. Cognitive processing is independent of these independent pathways, right? Second is that fully functional auditory and motor systems are not necessary conditions for the emergence and operation of language capabilities in the left hemisphere, right? So, yeah. Uh, so even if my like Broca had said and Wernick had said, right? There was no loss of motor movements in the mouth, in the vocal cords, in the tongue. Still, the patient was aphasic, right? Or the patient could still listen to everything and understand probably everything. Still, they were aphasic, right? So that thing was not the problem. The problem was comprehension. So cognitive ability is not dependent on, or it is free of basically capabilities of fully functional auditory and motor systems that are present, right? Yeah. Sorry, did I turn this off again? Just a moment. Yeah. Well, spoken language represents only one of a family of language skills mediated by the left hemisphere, right? So there are multiple other things also that are there. Not every language processing is actually there in the left hemisphere, and we can read about them that in the upcoming slides, right? So this is just an overview. It is not 100% true and 100% proven. It is what has been linked, right? So logic, analysis, sequencing, linear, mathematics, language, facts, thinking words, words of songs, computation, etc., have been functions that have been linked to the left part of the brain, and creativity, imagination, holistic thinking, intuition, hearts, rhythm, non-verbal feelings, visualization, tune of songs, daydreaming, etc., are what have been linked to the right side of the brain. Some of these things, right? So, as we said, it is not only the left hemisphere that is involved in this kind of a communication, right? It could also be right hemisphere. So, what is the role of right hemisphere in aphasia, right? So, the damage to right temporal area corresponding to Wernicke's area. So, Wernicke's area and Broca's area are actually present in the left hemisphere, right? Corresponding to that, what is there in the right hemisphere? In the in corresponding to Wernicke's area, in the left temporal region leads if the area that is present in the right temporal area, if there is a damage to that area, it leads to disturbances in comprehending emotional aspects of speech, right? So over there, that is responsible for comprehending speech or comprehending the thing that is communicated to you, right? But if contrary to that area, what is present in the right hemisphere, if that area is damaged, then the emotional aspect of speech will be incomprehensible, right? Somebody might not be able to assess the remorse or the sadness or the happiness in somebody else's voice, right? For example, the ability to appreciate a person from a person's tone of voice, whether he is describing a sad or a happy event, right? That uh, ability might be lost, right? This is an actual condition that is there, right? Again, I have given a lot of references from Big Bang Theory yesterday also. And this is kind of a condition that is represented by Sheldon Cooper in Big Bang Theory, right? So that person is incapable of, identifying irony, remorse, sadness, or tone of people's voices, right? So this is due to failure of development of this particular area of the right temporal region, which is corresponding to the vernix area, right? So this is one condition that actually exists in people. So in contrast, damage to right frontal area corresponding to Broca's area leads to difficulty in expressing emotional aspects of speech, right? Now that also happens with people. Some people are physiologically incapable of expressing their emotions, right? That could also happen. Some people like that is because this particular area might not be developed very well or might be a little damaged, right? So this is the role of right hemisphere in aphasia. Thus, some neurons needed for language also exist in the right hemisphere, right? There is now considerable evidence that an intact right hemisphere is necessary to appreciate semantic subtleties of language, such as irony, metaphor, wit, as well as emotional content of the speech, right? There is also preliminary evidence that ability to enjoy and perform music involves systems in the right hemisphere. So I hope that part is also clear. Right? So that brings us to the components of the nervous system. Let us just check the time once. Okay. So if I begin with the components of the nervous system, it will take me around half an hour. Uh, so I am thinking about postponing this and taking it up with the structure of the brain. It will actually make much more sense over there also. What I am going to teach in the next session for this part, that I can just briefly go through, right? And I will actually teach it in the upcoming session, maybe, right? So the major components of the nervous system that I was going to teach was the CNS and the PNS, right? The central nervous system and the peripheral nervous system, and the components, subcomponents of the central nervous system, as well as the peripheral nervous system, right? Because after this, I am introducing peripheral nervous system because that is also a part of the nervous system, and you should know about it if you are studying if you are studying neurobiology. But since after this we will deal mostly with this thing, the brain itself, right? Brain, neurons, and neuronal function, etc. That is why I want to get done with or the introduction of this, so that even if slight references to PNS comes, you are aware of what it is, right? So that is what I was going to teach, right? This is the that part. So I'm not taking this up right now. It is not very long, but along with discussion, it will be at least half an hour, at least, right? So we can carry it forward to the next session. We are done with the historical perspective and with the origins of neuroscience, and a little bit introduction I have given into some case studies also, and into some methods that are currently used for assessing the brain as well, right? So, yeah, with that, I can just stop sharing the slides, I think. Yeah. So to those of you who were actually a part of the session, I hope you did understand it. If there is any confusion or doubt, you can let me know. Um, if not now, there is also a Google group that is available. You can always, share your perspective or your questions or queries in that as well. I will try to take it up either over there or in this lecture itself. I would personally encourage this to be a more interactive session, right? So I am not a professor, right? I am a scholar, well, as are you, right? So I think I'll be able to answer queries if you put them up across, if you put them across, right? So, like, I'll be able to answer them in a more simplistic manner. I will be able to understand your viewpoint, I think, much better as of now, right? I'm closer to your age. And uh, and thus, I would like it to be an interactive session because that will help you also, right? This way, I will teach, I will be done with my course. It really wouldn't matter much to me. But if you are putting up questions, maybe I will also get a chance to learn, because maybe I have to look up how to answer that, or how I should, convey to you that particular concept. Maybe I have not done it up to what was required right now. Maybe I can also improve upon that. So, if it is more interactive, it will help both of us, right? Otherwise, it is just a lecture series. I can just go on like that. it will not be much of a difference to me, right? So yeah, that is it. I hope that you understood what was taught to you, and we can meet in the next class, right? I forgot to stop the recording. I'll stop the recording and we can end the session.